Hello everyone. In this short video, let us learn the difference between osmolarity and osmolality. Both terms, osmolarity and osmolality, they represent the concentration of solute particles in the body fluid. Osmolarity refers to the osmotic pressure exerted by the number of moles of solutes per liter of solvent, whereas osmolality, it is the measure of osmotic pressure exerted by the number of moles of solute per kg of solvent. Both the words osmolarity and osmolality, they measure the osmotic pressure exerted by the solute particles, but the difference is in osmolarity it is per liter of solvent, and in osmolality it is per kg of solvent. So this you can remember, osmolarity contains R, so this R is there in the liter, so osmolarity is per liter and there is no R in the kg, so there is no R in the osmolality also. So osmolality is for kg. So this you can remember. Osmolality is more widely used than the osmolarity in the clinical practice. So let us learn osmolality of plasma more. So the osmolality of plasma is in the range of 280 to 300 milliosmoles per kg. So the unit of osmolality is milliosmoles per kg. As I said, osmolality is per kg of solvent. So how to measure this osmolality of plasma? It can be directly measured by an instrument called as osmometer, but in routine clinical practice it is usually the calculated one. Osmolality formula is 2 into sodium concentration 2 times potassium concentration plus glucose concentration and urea concentration in the plasma. So 2 times sodium concentration, 2 times potassium concentration, glucose and urea concentration. If you add all these 4 components, you will get approximate amount of osmolality. So this picture shows the different constituents of plasma which are contributing to the osmolality. So if you see, the major contributor to the osmolality is sodium, which is responsible for 135 milliosmoles per kg of osmolality. So same amount of sodium associated anions will also be there. So that's why we take double of sodium concentration. The next highest concentration is of potassium, which is around 3.5 milliosmoles per kg. The same amount of associated anions will also be there. That is the reason why we take double the amount of potassium while calculating the osmolality. Similarly, the other important constituents contributing to the plasma osmolality are calcium and associated ions, magnesium and associated ions, glucose, urea and protein. So if you take the other constituents apart from sodium and potassium, the next highest concentration is of glucose and then it's of urea. So that is the reason why we include glucose and urea in the calculation of osmolality. Even though the other substances like magnesium, calcium and their associated anions contribute to the osmolality, relatively their effect on the osmolality is less. So in order to make it to simplify the measurement of osmolality, we consider only double the concentration of sodium, double the concentration of potassium and concentration of glucose and concentration of urea, which constitutes majority of the solute particles which are contributing to the plasma osmolality. Thank you.